Can we start now? 46 have come. It's still very less, but let's start, okay? Not increasing. In continuation, or it is just the what concise part, okay, of what we have discussed in the previous classes. So we'll discuss about the neurotransmission, okay. Majority of it I have explained already in the previous classes, but as such, what is this neurotransmission? Neurotransmission is nothing but a synaptic transmission, or it is a junctional transmission, or we say it as a neurotransmission only. Okay, so in neurotransmission, let me try with this slide presentation. Okay, so the synaptic transmission, if you are asked what is synaptic transmission, it is the junctional transmission or neurotransmission, it is all the same. Okay, so in this, what happens? A neuron transports its information by way of nerve impulse. Means the information is transported from one neuron to another by the way of nerve impulse. The neuron will be generating some nerve impulses, and with this nerve impulse, it is going to transport an information. Okay, the nerve impulses are transmitted from one cell to another at the synapses. Okay, so I don't need to explain here again. We have discussed all these points already in previous two classes. The movement and transmission of impulses at the synapse is called neurotransmission or synaptic transmission. The transmission at most of the synaptic junctions is chemical. So this is what most of the synapses are of chemical type, while some are electrical type. Okay, so in the chemical type of synapse, impulse in the presynaptic axon cause secretion of chemical substance called neurotransmitter. At some junctions, the transmission is electrical. That means some junctional transmissions or some synapses are electrical synapse, or the junctional transmission is of electrical type. And at a few conjoined synapses, it is both chemical and electrical. Okay, so nothing much to explain here. Okay, already we have discussed. So in the neurotransmission, or what we have discussed in the characteristics of a synapse, these most of the points we have discussed already, but some few points, okay, I will have added over here, in addition to that. And there is no harm that you add these points when you write characteristics of a synapse. No problem, okay? But when we discuss about the synapse, I have described only the salient points, okay? But when we are discussing about the neurotransmission, different stages, I'm describing little in detail. So in the stages in transmission, stages in neurotransmission at the synapse, okay? So this is a synapse, and here it is a presynaptic neuron, and this is a postsynaptic neuron part. Here it is written dendritic spine. That means this, and here it is written axon terminal. So this synapse is exodendritic synapse. So in this, here will be the synthesis of neurotransmitters in the presynaptic neuron. The synthesis, yes. Wait, huh? come? Have they come? Still problem is there or have they come? Yes, they have come? Or no, let them try again. I'm on the page, on that page, admit page. Let them try again. It's okay now. Okay. Okay. Yeah, I was telling about synthesis of neurotransmitters. Okay. So first of all, for neurotransmission, the first important thing is we need a neurotransmitter. A chemical substance called neurotransmitter is required. For that neurotransmitter has to be synthesized. The synthesis of neurotransmitters takes place in the axon, and during the whole process of synthesis, it comes down, okay, in the cell body, and then it comes down the axon, and then they are stored in the axon terminal. Okay, you know the cell body, the soma of the neuron. It here inside the soma of the neuron, there will be initiation of synthesis of neurotransmitter, and then by the time and slowly it will be transported, okay, from the body. It will be transported down the axon, and then during this process of the transport, it will get matured, okay, further matured, and then finally they will be matured and stored in the axon terminal, okay, in the form of vesicles. Okay, then the neurotransmitters are stored in the vesicles, okay, or they are simply the storage granules. So these neurotransmitters are visible here diagrammatically. I have shown here. So this is one axon terminal, and here it is having vesicles containing the neurotransmitters. Okay, so we call it as also synaptic vesicle having the neurotransmitters. Okay, so neurotransmitters here. Then when there is say like the passage of an action potential, okay, when the passage of an action potential down this axon terminal, okay, there will be increase in the influx of calcium. The calcium channels will open. Something like when the axon potential is generated, all the positive ions are the channels for positive ions are open. Okay, most exclusively it is for sodium and potassium, and to some extent there will be calcium also. Okay, so during this, calcium will influx. Okay, the calcium will enter the axon terminal. Okay, so by the time this calcium will be available in the extracellular area, because the calcium is having the calcium's concentration is higher extracellularly than intracellularly. Okay, so when the calcium channels are open, then the calcium will be able to enter this exon terminal. When calcium is higher concentration of calcium is present in the exon terminal, the intracellular concentration will increase. During this, there will be migration of the vesicles from this area towards the end of this exon terminal towards the synaptic site. Means slowly and slowly this vesicle will migrate towards the end. Okay. Then the membrane of the vesicle will come in close contact with the membrane of this exon terminal, okay, of the end button. Then due to this close contact, the membrane of the vesicle will be ruptured. Due to the rupture, along with the membrane of this exon terminal, due to this, there will be release of neurotransmitters to the extracellular space. Okay, so firstly, when there is change in the action potential in this, or when there is change in the resting membrane potential, then there will be entry of calcium ions from extracellular fluid to the inside of the exon terminal. Due to this, there will be movement of the synaptic vesicles towards the end of this exon terminal. The membrane of the vesicle will come in close contact with the membrane of the exon terminal, and there will be rupture of these, and it will release the neurotransmitter. Okay, so in the next step there will be release of neurotransmitter, and these neurotransmitters will find their receptors. The receptors will be present on these postsynaptic neuron. Then the neurotransmitters will bind to their receptors, and whatever the neurotransmitters left over here unbound to the, maybe due to the unavailability of the sufficient receptors, okay, they will be destroyed or they will be deactivated. There will be deactivation of the neurotransmitters either enzymatically or they may be taken back. Okay, they will be taken back into the terminal from which it came for reuse or degraded or removed. So some enzymes will be present over here. So, have you heard the name of the enzyme cholinesterase or acetylcholinesterase? So that enzyme acetylcholinesterase is an enzyme which can destroy, which can hydrolyze the neurotransmitter acetylcholine. Any excess amount of acetylcholine left in the synaptic cleft, okay, some have bound to their receptor and some have been left, okay, and those leftover acetylcholine will be destroyed by hydrolysis by acetylcholinesterase. Or this may again be there can be again reuptake. Okay, so these acetylcholine or any neurotransmitter, these will be again taken back up to these presynaptic neuron, and they will be again used up for resynthesis. Okay, so they will bind to their receptors. Leftover ones, they can be taken up or they will be destroyed. Okay, so these are the stages in neurotransmission at the synapse. So if those enzymes which can break down those neurotransmitters are not available, in that case these neurotransmitters will wait. They will wait for another time. Okay, they will wait quite long time if they don't if further they cannot bind, then they will be destroyed. Okay. So these neurotransmitters, they can be broadly classified into 5 categories, and this classification is not fixed. Okay, it can be classified in many ways. In many other books, you will find in different other forms of classification. Okay, so whichever form you follow, there is no problem. So as per my classification, they are classified as acetylcholine. Example is acetylcholine, biogenic amines. Okay, so those extra neurotransmitters having amines like catecholamines, okay, dopamine, catecholamine, then dopamine, norepinephrine, epinephrine, serotonin, and histamine. So these are all having amines. Amino acid neurotransmitters like glutamate. So glutamate comes under an excitatory amino acid category, and gamma butyric acid. In short, we write GABA. This comes under inhibitory amino acid neurotransmitters. Okay. Then the neuropeptides. Okay, so these are small peptides and they act as neurotransmitters or neuromodulators. They may be endogenous opioids, oxytocin, tachykinins. Okay, so these are neuropeptides. Oxytocin is a hormone. They can also act as a neurotransmitter and they come under the category neuropeptide. Okay, some other miscellaneous category consists of some gases like nitric oxide. They also act as neurotransmitter, and purines. Okay, that is a adenosine and ATP. Okay, adenosine triphosphate. Okay, so these are the classification of neurotransmitters. So there is no hard and fast rule for classification of neurotransmitters, but the examples should be correct. Whichever form you follow, there is no problem. Okay, in some other books, there will be different way of classification. So in that, you can follow anyone, but when you classify them, their examples should fit into that category. Okay, so as per my classification, it is 5 types: acetylcholine, biogenic amines, amino acids, neuropeptides, and miscellaneous substances, miscellaneous neurotransmitters like nitric oxide, which comes as a gas, and some purines like adenosine and ATP. They also act as neurotransmitter. Okay, so as per for neurotransmission, do you need to ask anything regarding neurotransmission or neurotransmitter classification? Do you have any query? Okay, so I will continue to the next section. Okay, next topic. That is degeneration of nerve cells. Okay, there are 2 terms. One is degeneration of nerve cells, another is the regeneration of nerve cells. Okay, so how the nerve cells get degenerated means how they are destroyed or how they become degenerated. Okay, and how they can be regenerated. That is, degeneration of the nerve cells occurred by 3 modes. Okay, one is the anterograde degeneration, that is also called Wallerian degeneration. Another mode of degeneration is through chromatolysis, and another way is the retrograde degeneration. Okay, so anterograde degeneration or what Wallerian degeneration means when an axon is interrupted. Okay, the distal segment invariably degenerate. That means suppose the body or the soma of an neuron is interrupted. Okay, that means there is some injury. Then the distal segment, the axon or the axon terminals, they get degenerated. Okay, so this kind of degeneration is called anterograde or Wallerian degeneration. Another way of degeneration is through chromatolysis. If a peripheral nerve, okay, here we are talking about a peripheral nerve, okay, not the central nerve. If a peripheral nerve is crushed but not severe, you understand? Severe means complete injury, okay? Crushed means to some extent it is crushed, okay? Some injury is there. Then the cell body may undergo swelling and displacement of the nucleus. So in that case, the cell body swells up and there is displacement of nucleus. Okay, and that kind of degeneration is called chromatolysis. Another way of degeneration is retrograde degeneration. That means all the changes proximal to the lesion site. That means if the lesion is at the axon terminal and the degenerative changes are happening in the axon hillock or in the body of a neuron, then that kind of degeneration is called retrograde degeneration. Okay, so there are three modes of degeneration of nerve cells. One is anterograde, okay, another is chromatolysis, another is retrograde degeneration. Okay, so here what actually happens when the peripheral nerves are injured. When they are injured, the first reaction is that the cell body may undergo chromatolysis. That means the body, it swells up. Then the macrophages removes the degenerating axons and the myelin sheaths, leaving the Schwann cells. Here, the Schwann cells will help in the regenerating process. Okay, so initially what will happen when there is degeneration, macrophages will remove the degenerating axons and the myelin sheath, but the Schwann cells will be left. So after a period of days to weeks, the surviving axon begins to sprout out as guided by the residual band of Schwann cells. So these Schwann cells will guide in formation of new axons or new portions of a neuron. Then successful regeneration depends on the availability of neutrophils secreted by Schwann cells, and Schwann cells, and the full recovery may take for 3 to 6 months. If the regenerating axon fails to locate a band of Schwann cells, then the outgrowing sprouts may end up blindly in connective tissue, forming a neuroma. That means a scar will be present, but not the this will not be able to bring back that that portion of that neuron, maybe the axon, the dendrite, or the spine. Okay, so the outgrowing sprouts may end up blindly with connective tissue, forming a neuroma. That means if Schwann cells are not enough, if Schwann cells do not help in the regenerating process, then it is going to lead to a neuroma and there will be formation of a scar. But in case of the central nerves, okay, when they are injured, the astrocytes and microglia invade the injury site. Okay, when the peripheral nerves are injured, there will be chromatolysis. Okay, and there will be the macrophages will remove the degenerating axons and it will leave the Schwann cells. The Schwann cells will help they will guide the regenerating process. And whole of this regenerating process depends on the availability of neutrophils and the Schwann cells. If they are not there, there will be formation of just a scar. It will not recover. But in case of the central nerve, when the central nerve is injured, astrocytes and microglia, they are present in the where? In the secretion of the ependyma in the where in the cerebrospinal fluid. So they will invade the injury site and they will remove the any toxic substances or any poisonous organisms. Maybe it is a bacteria, maybe it's a virus, whatever. Okay, so the astrocytes and the microglia will try to invade the injury sites. The oligodendrocytes and vascular pericytes will become very reactive, and if there is capillary damage, then there are large number of blood-borne phagocytes will come into the injury site also. Means if blood vessels are torn. Okay, if not, the astrocytes and the microglia will take care. Okay, so when the debris has been removed, the astrocyte processes will fill up the empty spaces, forming a dense glial scar. That means due to the migration of more and more astrocytes to kill the invading microorganism at the site of the invasion or the infection, then they will proliferate, they will fill the empty spaces, and they will lead to the formation of a glial scar. Then what will happen? Side by side, the oligodendrocytes from the myelin and segments around the masses of cellular debris. So there will be also initiation of more and more oligodendrocyte formation, and these oligodendrocytes will form the myelin and segments around the masses of cellular debris. That's how the central nerves will respond. Anything? Any clarification is required up to this? Hello?
No, ma'am.
Then next part is about the different functional areas of the brain. So these functional areas, you can find it out from the from the Google also, or otherwise also my notes are also having these functional areas of the brain. Okay, so simply you have to remember. Let's say some important functional areas I have listed out here. Say the somatosensory area. Okay, somatosensory area, it is located in the parietal lobe of the brain. General motor area located in frontal lobe. Premotor area in the frontal lobe. Association area in the frontal lobe. Visual area occipital lobe. Auditory area occipital lobe. Then speech and smell also in the frontal lobe. Okay, so that is one is auditory, speech and smell. Okay, so these sites you should get familiar. And yes, and different portions of the hypothalamus. Okay, I'm coming into the brain. Different parts of the hypothalamus. Say, hypothalamus is having different locations, and the different locations are having some similar types of groups of neurons, and those types or groups of the neurons have been termed as a nucleus. Okay, and different nuclei are present in the hypothalamus. One is lateral hypothalamic nucleus or medial hypothalamic nucleus. Okay, like that. Then ventromedial hypothalamic nucleus. lateral hypothalamic nucleus. Okay, this medial is extra over here. Anterior hypothalamic nucleus, lateral and posterior hypothalamic nucleus, suprachiasmatic nucleus. Okay, so particularly in case of the mammals, this is playing important role in case of the mammals for maintaining circadian rhythm. Paraventricular nucleus, supraoptic nucleus, diffusely located parvocellular hypothalamic neurons. That means diffusely located means they are all scattered. Okay, so this nucleus is associated with hunger and thirst. Lateral is stimulation of the lateral hypothalamic nucleus will lead to satiety, and the ventromedial hypothalamic nucleus stimulation will lead to I think there is a mistake. Ventromedial nucleus should be associated with satiety, and lateral should be associated with just 1 minute. Okay, there is a mistake, I feel. Let me check once. Typing some mistake is there. Ventromedial nucleus is satiety. Lateral hypothalamic nucleus is hunger. Wait. Stimulation. Yes. Okay, so I'll change this one. Okay, lateral hypothalamic nucleus. This should be medial. Medial hypothalamic nucleus is for satiety, lateral hypothalamic nucleus is for hunger, and ventromedial hypothalamic nucleus is for satiety. Then ventromedial nucleus is for satiety. If it is like this, you change in your notes, okay? I'm not sure how it is given in your notes. Lateral hypothalamic nucleus is associated with hunger and thirst. Do you hear? Can you hear?
Yes,
okay.
ma'am.
You remember that lateral hypothalamic nucleus is associated with hunger and thirst. Medial or ventromedial hypothalamic nucleus, okay, it is associated with satiety. Means stimulation of this portion of hypothalamus, ventromedial hypothalamic nucleus, will lead to satiety. Satiety means you feel full, you don't feel hungry, okay? And hunger, and when the lateral hypothalamic nucleus is stimulated, it is going to lead to hunger. Then anterior hypothalamic nucleus, it is associated with temperature regulation. This is say why, you know, when many a times when you have fever and all, as a practice, we used to put cold water on our forehead, okay, because this portion of the hypothalamic nucleus is towards the anterior hypothalamic nucleus. Okay, that's why we try to cool down. Okay. You remember like that. Okay. So anterior hypothalamic nucleus is associated with temperature regulation. Lateral and posterior hypothalamic nuclei, they are associated with cardiovascular function. And suprachiasmatic nucleus, okay, that is SCN. In short, we write as SCN, and this is in case of the mammals, particularly. Yeah, I write here. Rhythm in mammals. Okay. In case of the other non-mammals, okay, the other animals, not mammals, this circadian rhythm. Do you know what is circadian rhythm? Any idea? What do you know about circadian rhythm? Do you know, have you heard this term, circadian rhythm? What do you in this?
Ma'am, due to day and night sleep, ma'am, due to melanin, ma'am, 24-hour cycle.
Like any activity, okay? Any activity which repeats in the cycle of 24 hours. Do you get the point? So any activity which repeats in a cycle of 24 hours in a day. Say today at some 2 o'clock, one activity is happening, and that activity will happen again tomorrow at 2 o'clock in our body. And who is controlling that? That is controlled by the suprachiasmatic nucleus. Do you get something like you open your eyes, okay, after you sleep, you open next morning, you open your eyes and get up at some particular hours every day, right? You don't get up, you again sleep it, that is another. But you open your eyes, you wake up, right? You wake up and then you open your eyes, and it is told in your mind that yes, it is morning. Okay, so there is a biological clock in our body, that is the suprachiasmatic nucleus in case of the mammals, and this is the suprachiasmatic nucleus who is going to control any activities who are repeated in the cycle of 24 hours. Okay, every day, which is going to repeat. Okay, so this is controlled by suprachiasmatic nucleus. Yes, melanin also has some role. Okay, the secretion of not melanin, melatonin. Secretion of melatonin, it also happens in a circadian pattern. Yeah, and that is again controlled by the suprachiasmatic nucleus. Okay, so how the melatonin secretion occurs, that we will discuss later. Okay, when we discuss about the circadian rhythm, all those things in detail. Okay, so simply you know what simply you know the meaning of circadian rhythm. Circadian rhythm is any activity which is repeated in the cycle of 24 hours. That means today, if it is happening at 1 o'clock, it is going to happen after 24 hours at 1 o'clock. Again, after 24 hours at 1 o'clock. This such kind of rhythm is a circadian rhythm. Okay, say biannual means twice a year, 2 times in a year. Circadian means it is in a cycle of 24 hours. Another paraventricular nucleus, this is associated with oxytocin and a vasopressin release, and also male sexual functions. They are controlled by paraventricular nucleus. It is in short written as PVN. Okay, and supraoptic nucleus, that is SON. You may be asked the full forms. Okay, you try to know these full forms. So I may give as SCN, you may be asked to write the full form. Or I may give PVN, what it stands. Or I may ask that where is process SCN associated, where is PVN associated, or where is SON associated. So you are supposed to know the full form. Okay, and the supraoptic nucleus, it is also associated with oxytocin and vasopressin release. Here, 2 points are there: this paraventricular nucleus and the supraoptic nucleus, those nerves, okay, present in the paraventricular nucleus or supraoptic nucleus. They have two types of nerve cells. Two types of nerve cells. One type will secrete oxytocin, another will secrete vasopressin. SON also will have two types of the nerves. One will secrete oxytocin, another will secrete vasopressin. Okay, so paraventricular nucleus is associated with oxytocin and vasopressin. Supraoptic nucleus is also associated with oxytocin and vasopressin. Their locations are different, but their functions are almost the same. And other diffusely located paraventricular hypothalamic neurons. Okay, and they project to the infundibulum and release the releasing and inhibitory factors in the hypophyseal portal system. So these, when you discuss about the endocrinology, when you discuss about the releasing hormones or the inhibitory hormones, okay, synthesized in the hypothalamus are transported, okay, they are transported down the through the infundibulum, okay, and they will be stored or they will be released into the hypophyseal portal system. That means a set of capillaries which are running in and around the hypothalamus and the pituitary. Okay, those substances, those releasing and the inhibitory factors, say like GnRH or corticotropin releasing hormone, or what else? Thyrotropin releasing hormones. Okay, these kind of the hormones, releasing hormones or the inhibitory hormones, they will be synthesized in the parvocellular hypothalamic neurons, and they will be projected through the nerve tracts, and they will terminate in and around the hypophyseal portal system. They will pour their secretions into the capillaries present in the hypophyseal portal system, and from those capillaries again will get the pituitary will get those releasing or inhibitory factors or the hormones. They will find their target organs to the target receptors to the anterior pituitary or to the posterior pituitary. Okay, so up to this, is it okay? So in today's class we have discussed about neurotransmission. What is degeneration of nerves? What are the different patterns, different modes of degeneration of nerves? 3 modes of degeneration we have discussed. Another is regeneration of nerve, we have discussed. Another is functional areas of the brain, we have discussed, and different nuclei of the hypothalamus. This we have discussed. 5 points we have discussed. Okay. I will not continue more. You read this in detail. Okay. It must be there in my notes. And this part you correct it. Lateral hypothalamic nucleus. Okay. So this is associated with hunger and thirst. Ventromedial hypothalamic nucleus is associated with satiety. These 2 points you correct it in the notes. Do you want to ask anything? Yeah.
Excuse me,
You want
ma'am.
to ask anything? Yes.
Ma'am, could you please explain the reaction of peripheral nerve to the injury, ma'am? That chromatolysis part, ma'am.
Okay. Actually, I have not gone much into details of the chromatolysis process. Okay. Anyway. That is somewhat complicated. I have not given much into details. This degeneration of nerves, the chromatolysis, you mean?
No,
Here.
ma'am, the reaction, ma'am, of the peripheral nerve, ma'am.
Oh, this one. Here, the cell body means reaction of the peripheral nerve to the injury. That means these peripheral nerves means the nerves which are supplying to the peripheral organs, right? They are not in the central nervous system, right? And they are myelinated by Schwann cells, right? But in case of the central nerves, they are myelinated by oligodendrocytes. Yes or no?
Yes, ma'am.
Yes. Okay. So this difference you know it, that the peripheral nerves are myelinated by the Schwann cells and the central nerves are myelinated by the oligodendrocytes. Okay. So when these peripheral nerves are injured, then nearby these peripheral nerves there will be blood vessels. Or no? Yes,
Yes, ma'am.
because they are going to the peripheral tissues. So those blood or the tissues will be having the macrophages. Okay, so any injury to the nerve, they will be all nearby the tissue macrophages will come to that injured area because these macrophages are attracted to that injured area. Okay, so the macrophages will come to the site of the injury of the nerve and the macrophages will remove the degenerating axons and the myelin. Okay, and only few Schwann cells will be left. Okay, that majority of it will be removed by the macrophages. Then these Schwann cells will again regenerate. Okay, it means they will divide. Okay, so after a period of days to weeks, the surviving axon, the leftover axon will begin to sprout as guided by the residual vein of Schwann cells. So these Schwann cells, what they are going to do, they are going to nourish. They are going to provide the nutrition to this surviving axon. Do you remember when we discussed about the oligodendrocytes or these glial cells? Their main role is in providing the nutrients. Okay, so like that. So these Schwann cells, they will be helping the axon to sprout out. That means to sprout, you understand, to sprout out. Then so whether this process will be successful or not, it will depend on the availability of the neutrophils. So neutrophils, these are nothing but the nerve growth factors. So these nerve growth factors will be secreted by the Schwann cells. If these leftover Schwann cells are capable to secrete the neutrophils enough, then the regeneration process will be successful. If not, it may not be successful. Is it okay? If the injury was too much, if no Schwann cells are left, okay, and even if some are left, but these are not able to synthesize or produce their nerve growth factors, the Schwann cells may not be able to guide or to nourish this leftover axon. Okay, so this full recovery may take from 3 to 6 months. So if the regenerating axons fail to locate, okay, so if these axons are not aided by the Schwann cells, okay, if the regenerating axons fail to locate a band of Schwann cells, the outgrowing sprouts may end up blindly in connective tissue. That means instead of becoming again a myelinated axon, it will turn into connective tissue, and it will lead to the formation of a scar. Okay, and that is called a neuroma. Some scar will be present. Okay, that is a connective tissue. Say, just like when we have skin injuries and there is a line visible as a scar. That is nothing but the connective tissue. So this connective tissue is filling up the gap between the two injured sides, okay, and it is filling that area. So that is called the scar in other tissues, and in this, that portion which is formed by connective tissue, this is the neuroma. Is it okay?
Yes, ma'am. Thank you.
Okay, but here, in case of the reaction of the central nerves, the process will be little different, because here the macrophages will not be available, but the astrocytes and the microglia. So these microglia are the resident macrophages in the nervous system. They will help in that process. Okay, and in that there will be no role played by the Schwann cells, but the role will be played by the oligodendrocytes. Okay, then can I end the class here, or do you still need any clarification? Anyone wants any clarification? You can ask. The attendance is very low, okay? It's not good, okay? Nervous system, you should try to attend the classes. If no more questions, then let's end here our class. Okay? You can leave.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.